Sure.
Speak. We are now on the 7, myself included, so...
Yeah, okay. We will. So, good morning, everyone. Welcome to today's guest lecture. So it is my pleasure to introduce Professor Joe Leroy, a leading expert in veterinary science with a remarkable career spanning over 2 decades. He's a ruminant veterinarian by training, and he has extensively studied the impact of metabolic health on fertility, oocytes, and embryo quality in dairy cow. Currently, he is a full professor at the University of Antwerp. Professor Leroy has authored over 150 peer-reviewed papers and has been invited speaker at more than 60 international conferences. So his research, teaching, and advisory role, alongside his leadership in assisting reproduction, make him a distinguished figure in veterinary and reproductive science. And please join me in welcoming Professor Joe Leroy. So today he is going to talk about maternal metabolic health and fertility, caring about mothers and daughters' oocytes. So the floor is yours.
Thank you very much for this very nice introduction. Can you hear me loud and clear? All set?
Yes, very well.
Okay, because Estonia is a long way, so sound has to travel digitally and come out clear out of your speakers. So first of all, thanks for the very nice invitation. It was a pleasure to be with you all in springtime, and it's there that we had very nice talks and discussions, and that we got the idea of organizing this seminar. So thanks again for the opportunity. What I'm going to do today is to share some of our results of let's say, the last 8 years. And I'm going to try and share my screen. Is this working well?
Yeah, that's fine.
All right, fantastic. Just try to put you back on my screen. There we go. So we're going to speak about some results of the last 5 to 8 years, and I'll take you through a story, stepstoning through all these results. So what I'm not going to do is dive into deep molecular pathway analysis. We do these type of research. However, I think for the purpose of today, it's nice to give you a more broader overview of what we did over the last years. And of course, if you have more questions afterwards, or you want to reach out and contact me or visit the lab, you are more than welcome to do so and to discuss about more scientific questions, or to dive into a very nice collaboration. So I see it as a kind of an introductory lecture. I introduced some challenging topics and challenging hypotheses and thoughts, just for you to challenge me again and just to stimulate the discussion. So, all right. So what I'm going to speak about, I'm trying to launch my laser pointer. Can you see the laser pointer? Yes. Okay. So we're going to speak about maternal metabolic health and fertility. And I changed the title just a little bit. And I set a subtitle about how should we care for the oocyte, but also about the oocyte, not only of the mother, but also of her daughter. So, okay. In this talk, I will try to cover 3 topics. First of all, why should we bother about oocyte quality? We all know oocyte quality is very important to guarantee fertility, maternal fertility, but I'll give some more insight there. And then I would like to expand a little bit on how long it takes before an oocyte is affected. I mean, imagine you change your diet as a mother. Do you have to be concerned within the 24 hours, or are you relaxed for the coming 4 months before, for example, you may expect impact on oocyte quality? So we're going to answer some straightforward questions there. And then, of course, when oocyte quality is studied, we always focus on the mature oocyte, or the oocyte just before ovulation or just after ovulation. However, we all know that there is a dormant follicle pool in the ovary, and I would like to expand a little bit on how this dormant follicle may be affected, yes or no, or should we consider them as a completely insensitive dormant follicle pool. And then, of course, as we are veterinarians, at least I'm a veterinarian, as said in the introduction, it's always nice to solve a problem. If there is a problem, it's nice to try to find solutions, right? So I would like to raise some concept. Is it possible to cure oocyte quality, or to change the maternal diet, for example, to re-establish oocyte quality, to recover oocyte quality, etc. So we're going to speak about some opportunities from in vitro applications, from feed bank applications, and there I'll try to refer a little bit to my dairy cow background. And then, of course, we will speak about preconception care opportunities of the mother. Who are the stakeholders of my talk today? First of all, livestock reproductionist. I'm a veterinarian in heart and brain. I am focused on dairy cow fertility. However, within my university, it's difficult to explain the concept of dairy cows because there are not a lot of veterinarians involved there. So I'm using my dairy cow model and other animal models more and more to learn more about general biology of the oocyte, fundamental biology of the oocyte, and also to somehow ship my ideas towards and the results towards human medicine, right? So second important stakeholder of the data I'm going to present today is human reproductive world, and we have very intense collaborations with these partners as well. So, okay, why should we bother about oocyte quality? First of all, so that's the oocyte. I hope you recognize this. I think it's the most beautiful cell of the world. This is the oocyte surrounded by cumulus cells, these somatic supportive cells. However, the oocyte is not an easy cell to study. It's completely different compared to somatic cells. So that makes it already difficult to extrapolate interpretations we got from somatic cell towards that female gamete. Of course, we all know the oocyte is the cornerstone of female fertility. No good oocyte is no fertile conditions. It's the source of new life. That's really important. Do not forget that all the mitochondria, for example, you have in your body are just inherited through the oocyte. So your father didn't add any mitochondrion upon conception. So all your mitochondria comes from your mother's side. So if something is wrong with your mitochondria, you should blame your mother and not your father. That's important. The oocyte, upon ovulation, the quality of that oocyte is the result of a very long growth path. Yeah, it takes weeks or months, depending on the species, before an oocyte is ovulated. So we may expect that during that growth path, there is a kind of a sensitivity to its environment, and I'll expand on that later on. So it's a very sensitive cell. We all know from in vitro cultures but also in vivo environments that if you change the microenvironment of an oocyte, that has direct, sometimes drastic consequences for oocyte quality. The oocyte is vulnerable. It's a sensitive cell. It is a plastic cell, but when you have a too high concentration of specific compounds, the oocyte will suffer, and Mother Nature will say, okay, that oocyte will lead to bad quality conditions, and it will hamper fertilization and post-fertilization development. So it's vulnerable. Oocyte quality impacts next generation, and we will also expand a little bit on that. I already said that mitochondria of an oocyte are the mitochondria that will be given to the next generation. So there's not only through mitochondria, also through other mechanisms, for example, DNA epigenetic modifications. What happens in the oocyte may drastically impact postnatal health. The oocyte is super difficult to study. It's far too big to nicely visualize under the confocal microscope. We all have to do tricks to make them nicely visible and studyable. So it's a difficult cell to study from several perspectives. And then, of course, should we care about the oocyte? And if we should care about the oocyte, how should we care about the oocyte? And secondly, should we care for the oocyte? Is it possible to develop care for the oocyte, and how should we approach that? And if yes, how should we do that? I just did a quick search on literature some days ago, and I focused on studies. We published some review papers in that perspective, but I just picked one of the latest review papers on that perspective. In an animal model or in a whole animal condition, can we improve oocyte quality? Many researchers try to focus on that question. So by, for example, changing the diet, supplementing the animal with all kinds of antioxidants or all kinds of supporting conditions, can we change oocyte quality? So, and Miguel Velasquez from the UK, he from Newcastle, he overviewed literature very lately, and he came up with a very simple conclusion. He said, you can do whatever you like in an animal model by supporting the animal through nutrition or whatever. It's not very clear what it does on oocyte quality. It's not straightforward, right? It's not like if you give metformin, that glucose tolerance drastically improves. That's a straightforward outcome, but that's not so straightforward in oocyte quality. And he also says if the mother is more or less healthy, nutritional impact is very difficult to discover in oocyte quality. And he also nicely explains the huge amount of confounders that are included in these type of studies. Most of these studies are underpowered and they are full of confounders because there are many factors impacting oocyte quality in vivo trials. Also, for example, very lately, some researchers from New Zealand discovered that oocyte quality is also steered through genetic progress. I mean, if you breed for genetic gain on reproductive outcome, they say that oocyte quality measures are also improving. So genetic background steering through genetic selection towards better reproductive outcome may have a drastic impact on oocyte quality. So genetic background is also important. But again, they also conclude if the animal is healthy and not being submitted to a physiological challenge, we don't see anything, right? So as long as the mother is okay, we don't see a lot of impact on oocyte quality. It's only when a cow is submitted to the physiological stressor lactation that we can see that a better breeding value for fertility leads to better oocyte proxies. So, okay. What about human medicine? As I said, we are also very interested in human fertility and then more specifically in human oocyte quality as a link between maternal health and maternal fertility. And what do we see there? It's nicely depicted here on this graph. So here on the x-axis, you see body mass index, and here you see the relative chance of a spontaneous conception. And you see with an increasing body mass index, you see that the chance for a spontaneous conception is significantly dropping. So higher BMI, lower conception rates. Very nice. But then my first question is, which was the research cohort that was used to do that epidemiological research? Well, it's very clear. This cohort was a fertile cohort. These were ovulatory women. The couple, the men within the couple had normal sperm measures as well. So these people were followed up, not for fertility reasons, right? They were just consulted in a pre-conception context. And in that healthy environment, they had a very clear relation between body mass index and the chance of conception. The question is, okay, this is relation, this is correlation, this is association, this is absolutely not a causative relink, right? It's not because we don't know whether body mass index has a direct impact on conception rates. That's an association. So is this a cause or a parallel observation? And then if fertility goes down, is this due to the fact that something is going wrong in these obese individuals with the oocyte? Or is it because the uterine environment is affected? Because becoming pregnant and staying fertile is much more than only the oocytes. It's a whole endocrine background, but also the uterus has to be receptive, etc. So it's not so easy to study. And then is the environment of the maturing oocyte affected? And if so, does this lead to reduced oocyte quality and thus subfertility? That's very difficult to answer that question in a human or animal model. For that reason, or a routine animal condition. So for that reason, we really need to build very strategically designed studies where we really use randomized controlled trials or really induce specific treatments and compare different groups. So, okay, we did that type of research. So we did a lot of explorations in human follicular fluid. So we produced some nice data. It's already like 10 years ago, by the way, where we analyzed follicular fluids of obese women and compared it with oocyte development capacity in vitro in a human clinical setting. So this is also an association study. And you see I sometimes use the wrong words in titles. Here we speak about consequences, which imply causation, while we only checked association. So it's important that we split causation from association. So we also analyzed fatty acid composition in follicular fluid, related it with fatty acid composition in human blood, just to see how the follicular microenvironment is eventually affected by the maternal health, by her diet, by her metabolic background. So we found very nice correlations and associations. But of course, is there a causation linked to oocyte quality? For that, it's important to use well-driven animal models or in vitro models. Why are we so interested in a maternal metabolic health to jump in these very specific research? As you all know, obesity becomes a pandemic, right? More and more people are affected by overweight or obesity. And if I just consult the latest data from the WHO, that's 2022, 2 years ago, they said that about worldwide, 1 out of 6 adults are obese, and about 20% of women at fertile age are obese. I'm not speaking about overweight, I'm speaking about obesity, so body mass index of 30 or higher, right? You see, if you compare that with 1990, at that time I was 13 years old. I think Alireza was a little bit older at that time already, but anyway, at 1990, only 7% of adults were obese, right? So there's a drastic increase with a drastic impact, the drastic impact on population health. So I'm not going to expound on the metabolic background and what happens metabolically wise and health wise in obesity, but if you have questions, please raise afterwards. But we know that adipose tissue is much higher in prevalence, right? These adipocytes will behave differently. You get a higher level of inflammation in these patients. These patients get a certain degree of insulin-reduced sensitivity, and we get higher concentrations of blood fatty acids, especially non-estified fatty acids. All of these factors are reflected in the microenvironment of the oocyte. Now that's clear. We did a lot of studies, a lot of other researchers did a lot of studies. So we know that that microenvironment of that maturing oocyte is clearly affected by the metabolic challenge that coincides with obesity. This question is evenly important, or this issue is evenly important in modern dairy cows. Modern dairy cows are selected to produce a lot of milk. However, due to selection for production of milk, they are really genetically pre-programmed to mobilize body lipids in the first weeks after calving. We call that a period of negative energy balance. And actually, when it comes to oocyte microenvironment, the consequences are more or less the same. They're also flooded by high concentrations of fatty acids, flooded by higher concentrations on inflammatory markers, etc., with downstream consequences for oocyte. So, of course, a dairy cow is not an obese person, but there are a lot of metabolic parallels that makes both situations very interesting to study when it comes to impact on oocyte quality. So, okay, we know if we focus now only on these fatty acids, that these fatty acids can be taken up by the oocyte. Let me take you a little bit through oocyte quality. So this is an oocyte. It's just a diagram of an oocyte. And I only depicted 2 main organelles, the nucleus and the mitochondria. Oocyte mitochondria are super important. There are about 100,000 mitochondria in oocyte, which is just incredible. These mitochondria behave a little bit differently. They look differently compared to a somatic cell. They are less mature, but they are absolutely crucial for oocyte biology. Why are they crucial? First of all, as you all know, mitochondria, they process substrates like pyruvate coming from glucose, glutamine, fatty acids whatsoever. Acetyl-CoA is formed, and then the mitochondria is processing these molecules in the Krebs cycle to produce ATP. You all know how important ATP is to just fuel all kinds of kinase processes. They are so important for spindle formation, for the chromosomal alignment at the metaphase, for the extrusion of the cortical granules, which is important for fertilization. So to make it very clear, people, women, or animals with defective mitochondria will have, for example, higher rates of aneuploidy, which is normal because ATP is lacking and then spindle formation is blocked or reduced with non-well synchronized chromosomal splitting, right? So that's a very important part. However, mitochondria, they do play also a crucial role in calcium homeostasis at cellular level. Right? We also know that mitochondria, if they work too hard, if they are submitted to too high substrate concentrations, they will produce too high reactive oxygen species. And you all know reactive oxygen species or ROS in several cell types. They are involved in several pathologies in cell types, and they may lead to damage to membranes and other stuff, right? Mitochondria are also a very important inducer of apoptosis. We call that the mitochondrial linked apoptotic pathway. That's an important one. And lately as well, only like the last 5 years, people discovered that mitochondrial functioning is directly causatively linked with epigenetic programming. Yeah? So all kinds of intermediates produced by the mitochondrium may have an impact on epigenetic programming of the nuclear DNA. So that's important. It's nicely described these days in somatic cells, but it's less studied in oocytes, and it is very important. So a lot of reasons to focus on mitochondria. And another reason, which I told you just in the beginning of my talk, these mitochondria will be transferred to the offspring, right? So if something then goes wrong with the mitochondria, it may have a long-term impact on offspring health as well. So that's an important point to consider. So we know if mitochondria are not happy, they are boosted. They are, for example, by the high fatty acid concentrations. If we expose an oocyte to high fatty acid concentrations, as in an obese individual, these mitochondria will work too hard, and this will lead to too high ROS formation, accumulation, and damage within the oocyte. So to make the story very simple, these fatty acids, for example, due to the maternal metabolic health conditions, will lead to reduced oocyte quality and reduced fertility. This link has been established, is well accepted, everybody knows. So I think that should be very clear. However, to really study what we know there from in vivo observations in the human and in vitro conditions, we of course need to apply that in very well controlled animal models. And for that we use mice. And here you see 2 mice, the inbred black 6 mouse. This is a control mice. This is an McDonald's mice, I say. So that's a mouse that was submitted to a McDonald's diet it's a high fat, high sugar diet, but we call it the McDonald's diet to wake you up a little bit. So you see the weight increases with 25%. We also use the outbred mouse, which is a Swiss mouse. And you see that Swiss mouse also explodes more or less if we expose it to 6 weeks of high fat, high sugar diets. Why do we combine both or started with combining both strains? Because we discovered that inbreeding as such has a drastic negative impact on oocyte mitochondria. So if we want to have a nice animal model studying diet and obesity on the impact and trying to discover the impact on oocyte quality, of course, we want a healthy mouse model with no innate mitochondrial defects. I will show you. So what happens if we feed these oocyte the McDonald's diet? You see that especially the abdominal fat drastically increases. And you see here the weight, going up. So the black lines are the black 6 mice and the gray lines are the Swiss mice. So both strains are nicely reacted on that obesogenic or high fat, high sugar diet. What does that mean for oocyte quality? Well, let's focus, for example, first on oocyte lipid droplets. And here you see some nice pictures where we had the fluorescent stain or the lipid droplets. So here you see the Swiss control oocyte, and here you see the Swiss high-fat, high-sugar oocyte after 6 weeks of McDonald's diet. So lipid droplets drastically increase. They almost double. It's very easy. It's very straightforward. Lipid content goes up, doubles in oocyte after a McDonald's diet for 6 weeks or 8 weeks. You see the same in black 6, but if you look carefully on the morphology of these lipid droplets, they are completely different. So there we see big clumps of oocyte droplets. They aggregate together with mitochondria. They look degenerated. So there, just by watching lipid droplets, we see already a big difference between oocytes of different strains. The Swiss oocyte much better resembles human oocytes, so that's already very important. We also checked mitochondrial abnormality. And look at this. I will focus first on the black 6, the inbred strain. So here you already see that the proportion of abnormal mitochondria in this oocyte is as high in the control group and in the McDonald's group, if I may say it like this. Yeah. So there we have already a big issue when it comes to mitochondrial aberrations in the control black 6. So black 6 for us is not a good model when it comes to oocyte mitochondrial features. You see here in Swiss oocyte, after 6 weeks of a high-fat, high-sugar diet, the proportion of mitochondrial aberrations, abnormalities morphologically within an oocyte is drastically increasing. So also that is very straightforward. If you give a mouse high-fat, high-sugar diet for several weeks, 6 to 8 weeks, we see that lipid droplets drastically increase and mitochondrial abnormalities morphologically based on transmission electron microscopy are also drastically increasing. Very straightforward. I think there's no room for discussion there. I will quickly show you that we also, of course, not only study morphology, but we also study mitochondrial function. And I'll try to show you how we do that on the slides. I think, of course, you know these procedures. So we stain the oocytes with GC1, and GC1 accumulates in mitochondria in the inner membrane space, where if a mitochondrium is very active, with a high inner membrane polarity, a high proton gradient, these mitochondria will have a yellow color, right? While mitochondria that are rather inactive have a greenish color. And in that way, we may estimate mitochondrial activity. If mitochondria are too active, they will produce a lot of ROS, and ROS are stained in red. So here we have inactive mitochondria, active mitochondria, and ROS formation. These are 3 images, 3 filters from the same oocyte, right? Okay. So this is a control oocyte, and watch carefully what happens if we take an oocyte from an obese mouse submitted to a high-fat, high-sugar diet. This will happen, right? So you see that the amount of ROS formation is drastically increasing, leading to an uncoupling. Mitochondria react on ROS formation and try to reduce membrane polarity. They will reduce proton gradient, leading to more inactive mitochondria, and you see that the amount of active mitochondria is drastically dropping. Yeah. Of course, this will lead to oocyte dysfunction. Of course, this will lead to final maturation issues. Of course, this leads to fertilization issues. Yeah. So that's basically what happens. What we also do in the lab, we measure oxygen consumption of oocytes. We measure ATP production, etc., with a seahorse bioanalyzer. So we do that as well with oocytes, embryos, etc. Okay, so that's a first thing. I will be quicker now. Second question, we know that oocytes are sensitive to their environment. We know that obesity or a lot of lipid mobilization or a wrong diet has a drastic impact on oocyte quality at a mature oocyte, at ovulation. But how long does it take to affect an oocyte? As I said, we know that after 4 weeks or 8 weeks feeding a high-fat, high-sugar diet, we see, for example, lipid droplet content in oocytes increasing. That's what I just said. We repeated that experiment. It's a PhD student that just finished her PhD and defended. Carlene Morkens, she defended in June. And she asked us the question, how long does it take? If we have a mouse just submitted to the high-fat, high-sugar diet, do we see already something after 24 hours? Or after 3 days or after 1 week? Yeah. And that's the result. Yes, we do see a drastic impact already after a very, very acute feeding regime. So going to McDonald's for 1 day leads to, and we were very surprised, at least in mice, leads to a drastic increase of triglyceride content in the oocytes. And after 3 days, 1 week, 4 weeks, 8 weeks, this is all published. But this is astonishing. We were the first to show that acute effect compared to the chronic effect. So it's not innocent. Diet has a direct impact on oocyte quality because after 24 hours or 3 days, mice are not yet obese. Right? They are just submitted to a different diet composition, right? So we are doing more trials in that direction. So it's not only obesity, it's also direct impact of diet ingredients that has an impact at oocyte quality. What about mitochondria? Well, here we estimated the proportion of abnormal mitochondria in oocytes collected from mice at 24 hours, 1 week, 4 weeks, 8 weeks, 12 weeks, and 16 weeks after the onset of feeding. And you see, it takes about 8 weeks before we see the proportion of abnormal mitochondria going up, right? So we don't see anything after 4 weeks so far, morphologically. However, there is one big confounder here, and that is age, because we know advancing age has a negative impact on oocyte mitochondrial features. We know that aged women 40, 41, 42 have reduced oocyte quality. That's generally described, nicely described in literature. And one of the issues is there that mitochondrial features are going down, right? So we corrected for that, of course. So I'll show you what the control group mitochondrial feature does. So they do increase a little bit with increasing age. So that's the light blue bars. However, we see there's stays a drastic impact of obesity. So at 8 weeks, mice are obese. So there we see an impact of obesity on mitochondrial features. So take it together. It goes really quick. We see lipid droplet content going up already after 24 hours. Mitochondrial features start deviating after 6 to 8 weeks of feeding. Okay. We can nicely mimic all this in vitro. I did it during my PhD, which is a long time ago. I defended my PhD in 2005, so that's almost 20 years ago. And Dumini said in the introduction, 2 decades of experience. So yeah, that's almost 2 decades. So what can we do? We can mimic the last follicular phase, the last maturation phase in vitro. We take an oocyte from a slaughterhouse ovary, we put it in vitro, we mature it. You all know the system for 24 hours. And during that maturation, we can add a lot of stuff we want to study. So we did that a lot of times by adding non-estified fatty acids. I explained to you the importance of these non-estified fatty acids. We added during oocyte maturation, and we checked zygote quality, morula development, and blastocyst formation. And you see all the outcome that has been affected. So it's very, very clear. Just by adding for 24 hours high fatty acid concentrations, mimicking what happens in obese women or in a negative energy balance dairy cow, we have drastic impact on development. We have a drastic impact on mitochondria. We have a drastic impact on lipid content of the oocyte. We see after fertilization in a routine culture environment, reduced morula development with genetic, metabolic, lipidomic deviations. And we also know that DNA methylation profile is changing. So adding fatty acids alters DNA, global DNA methylation profiles in a morula. And that's quite drastic. So, of course, then you can say, yeah, Joe, I mean, they look different, these blastocysts, these morula, after 24 hours of NIFA maturation, but who cares? Probably these embryos, once they are in a healthy uterus, they don't give a shit and they just develop, right? So we wanted to know that. What did we do some years ago? We went to the slaughterhouse, matured oocyte to control environment or to our lipotoxic, so the high NIFA environment. We fertilized these oocytes. We cultured them routinely, and we collected day 7.5 blastocysts from the well. And there we only selected the best blastocysts that were formed in vitro, right? So we did a kind of a, I call it a restart at day 7.5. Only the beauties in both groups were selected for transfer in a recipient cow. So the hypothesis was, if you transfer a beautiful embryo at day 7.5 in a healthy uterus, the history of that embryo is not important anymore. So that's what we did. We transferred these embryos coming from two groups, the NIFA maturation group or the healthy maturation group, in synchronized cows, and we collected the embryos. Normally at day 14, an embryo should look like this. So a nicely elongated spaghetti-like embryo. So at day 14, we collected the embryos to scrutinize embryo disc development, to see what the extra embryonic disc was doing, how big the embryo was. We checked gene expression patterns. We checked epigenetic profiling of extra embryonic tissue and the embryonic disc. We did interferon tau measurements, etc., etc. So we did a lot of outcome parameters. What did we find? So here people and students are helping to transfer these embryos and collect these embryos. Look, embryos coming from controlled matured oocytes, let's say 3/4, so 75% of these embryos were in the normal shape. They were tubular. Yeah? Only 1/4 was ovoid, and only the minimum was in a spherical, underdeveloped state. Look what happens if embryos originated from oocytes matured under high NIFA concentrations. Development was heavily jeopardized. Here we see a lower number of tubular, more spherical, and more ovoid. So these embryos are retarded in development. While we transferred them in exactly the same condition, we only selected top-looking blastocysts. So here we can nicely confirm that the blastocyst, even if she looks as a beautiful species, if she originates from a bad maternal environment as an oocyte, there are drastic downstream consequences for further development. By the way, it's not on the slide, but these embryos from the lipotoxic group, they were less capable of producing interferon tau. And you all know from physiology how important the interferon tau is for pregnancy recognition. So, okay, taken together, and that's a scheme I always show to veterinarians and to farmers. So this is the preovulatory oocyte. The oocyte is ovulated, comes into the oviduct, and is then fertilized and becomes an embryo. We know that high NIFAs or other metabolic issues, lipopolysaccharides, tumor necrosis factor alpha, all kinds of adipokines, oxidative stress measures, due to unhealthy maternal health, is reflected in the blood, is reflected in the microenvironment of the oocyte, and has a drastic, acute impact on oocyte developmental capacity and oocyte quality, mostly through affected mitochondrial features. This is as important for human medicine, important in livestock. It's difficult to study it in these animal or living conditions. So for that, we use specifically designed animal models or in vitro models. Okay, what about the dormant follicle pool? Yeah? You all know how ovarian physiology is going on. I mean, you know that before you reach an oocyte at ovulation, it's, at least in women or cows, it takes about 3 to 4 months to reach that pre-ovulatory phase. I hope you know that. So that dormant follicle pool is just there. Women are born, young ladies are born with hundreds of thousands of dormant follicles. They stay there. A lot of them just go in atresia, go in apoptosis, but some of them are activated after puberty, onset of puberty. So the question is, of course, does obesity affect the dormant follicle pool? Do we have to consider them? Do we have to think about them? Are they sensitive, yes or no? And if yes, does this have long-term impact on oocyte quality upon ovulation? So that was a question that was answered by one of my PhD students who is going to defend in December. So I will just show you these dormant follicle pools. So this is transmission electron microscopy pictures, and this is a nicely primordial follicle. So here you see the oocyte nucleus, here you see the ooplasm, right, with some black dots. These are the mitochondria, and then you see some flattened granulosa cells. That's a primordial dormant follicle. We always consider them as insensitive, difficult to study. Do we care? No, we don't care. They are there somewhere, and we don't think they feel what happens with maternal health. This is an activated follicle. So this is a primary follicle. So here you see the oocyte, you see these mitochondria, you see the nucleus, and here you see the cuboidal. So they are activated granulosa cells. They become cumulus cells. And then here you have a secondary follicle, as you all know, a nice oocyte with 2 layers of granulosa cells. So we studied these dormant follicle pools in mice. So what did we do? We gave mice a high-fat, high-sugar diet. We not only investigated the ovulatory oocyte, but also did a lot of research on the dormant follicle pool. And I will show you some nice things to know. For example, the right picture is a transmission electron microscopy picture of a mature oocyte. So an oocyte that has been ovulated. And you see these mitochondria, they look like this. They look immature. It's generally known that they look immature and they look quite dense. And the density refers to their activity. So while they are quite immature, they are active in a mitochondria, in an ovulated oocyte. That's necessary to fuel all these energetic demanding processes. However, if we compare that with mitochondria in a dormant follicle, so a primordial follicle, they look completely different. So you see these mitochondria, they have these nice few cristae, they look like Easter eggs. They are nice translucent, very nice mitochondria. They are less active, they are not inactive, but less active, so they look completely different. So what did we do? Again, only with the Swiss mouse, because we don't believe the inbred mice anymore, and I explained you why. We made them obese, so here you see the consequences, and we focused on mitochondrial features of the dormant follicle pool. So this is the dormant follicle pool, and all these follicles, more than 20,000 in total, sorry, all these mitochondria, so more than 20,000 were scored by my students in a blind way. And we calculated the proportion of abnormal mitochondria. And you see, or here is the proportion of normal mitochondria within these follicles. And you see that the proportion in primordial follicle is drastically reducing due to an obesogenic diet. So the results were subtle, but the differences are straightforward. So this means that an adult mouse being submitted to a high-fat, high-sugar diet, McDonald's diet, not only shows drastic deviant features in the ovulated oocyte, but we have clear signs of an affected dormant follicle pool. These primordial follicles show deviant morphological features within the mitochondria. And then the next question, of course, is imagine a female offspring is born from an obese mother, because all these dormant follicles are present already at birth, which is not completely true in mice, but in human, that's the case. Every young lady is born with all the follicles present in a dormant follicle status. So our question was, imagine we focus on these newborn pups and we focus on the dormant follicle pool. If that pup is born from an obese mother, do we see already affected primordial follicles? And that's what we investigated. So what did we do? Within 24 hours after birth, these pups were sacrificed and we isolated the ovaries. It is a challenge to find the ovaries within a newborn pup. It's tiny, 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 but Ine, my PhD student, she did a wonderful job. So here you see a kidney. Here is the uterine horn. There is another uterine horn. And here, I think you have to accept it because it's difficult to see, is a tiny ovary, right? So these ovaries were resected and we did a transmission electron microscopy, but we also did immunofluorescent staining. So we stained these very small ovaries for a lot of mitochondrial parameters. So PGC1 alpha, for example, is a regulator of mitochondrial biogenesis. TFAM is a DNA binding protein, so it stimulates, it activates mitochondrial DNA transcription. So, and many others. So all these factors were analyzed in these newborn ovaries. So here, for example, you see an ovary stained for TFAM, and then with image analysis, we evaluated per follicle, per primordial follicle. So every region of primordial follicle was identified, and then we try to estimate the amount of TFAM present. And what did we see? Very consistent. We see that pups at birth have a significantly reduced TFAM expression within the primordial follicles when that pup is born from an obese mother. And we see the same after weaning. So if we follow up the pup 3 weeks throughout lactation, that is consistent. So for the first time, we can say that the dormant follicle pool of a young lady, a baby born from an obese mother, is already affected and probably explains what we see at adult age within the primordial follicle pool, but also at adult age at the ovulatory phase. A last experiment that I want to show you is we wanted to know if your mother is obese and you continue as an offspring on the obesogenic diet, what it does with oocyte quality. Do we have an additive effect? Obesity mother, you continue as an offspring on that same diet, or if you change to a control diet, whether these features disappear, yes or no. The other way, if we do a crossover design, a 2 × 2 factorial design, how maternal background and offspring diet have an additive or a compensatory effect. That's what we investigated. And that's, by the way, the PhD thesis of Ine. So this is the experimental design. This is the maternal diet during pregnancy and lactation as well. And pups were then crossed over to 2 different diets. You see a lot of outcome was evaluated. I'm just showing you one outcome based on oocytes ovulated at adulthood in the offspring. You see that if you have an obese diet in the offspring, you see abnormalities in mitochondrial features goes up. That's what I already explained. But you see that there is an additive value here of maternal obesity. So if the mother is obese and as a pup you stay on that obese environment, you see an additive impact on mitochondrial features, negative impact on oocyte quality at adulthood, right? So that's important. That is all published. So if you want to dive into that experimental design and try to understand the data, please send me an email and I'm happy to send you the raw data sets if you like, but also the publication. Okay, very quickly to finish up, do we have opportunities to care for the oocyte? We know that the oocyte is suffering from its environment. It's sensitive. Can we apply something to recover? Yes, we can. We did a lot of studies by adding insulin transferrin selenium, for example, to see if we can boost these mitochondrial features, if we can boost development. Adding ITS, by the way, boosts development, but does not improve embryo quality. So take care. Numbers do not say everything. We also worked with antioxidants to see if we can cover up for the reactive oxygen species, and it works, but it's not easy. So sometimes you have to preload the oocyte with antioxidants to make her more robust to negative energy balance, or you have to recover afterwards. So it all depends on the molecule, how the best way what the best way is to use it. Lately, we published quite some work using MitoQ. Mitoconic acid is not mitoconic acid. MitoQ is metoquinone, is an antioxidant that targets mitochondria, right? So it's really the molecule of choice to support mitochondrial functioning. And yes, we see that if you have an oocyte maturation under lipotoxic conditions, and then you try to recover during culture with MitoQ, we can completely restore embryo development, but also embryo quality. So here, this is the control group. Here is the lipotoxic group, so matured under high palmitic acid conditions, and then just cultured under control. You see embryo development is drastically dropping. However, if we add MitoQ during culture, these embryos, they recover. Yeah, so it's a kind of a backup, a kind of a therapy. Yeah? They do in terms of development, they do in terms of quality, but they also do in terms of gene expression patterns. So you see genes related to oxidative stress, mitochondrial stress, and endoplasmatic reticulum stress are somehow completely recovering when we add MitoQ in the culture environment. So that paper was published in Human Reproduction because, of course, the human medicine is also interested there. We can also add mitoQ during maturation. That's absolutely not a problem. We can add it during maturation, and there we can protect the oocyte from high NIFA concentrations. So, okay, are there opportunities from the feed bank? Can we add something to the diet of the dairy cows? Yes, we can. Based on a field trial we did, we know that most of the dairy cows around, and I will be a little quicker here in terms of time, the dairy cows early postpartum, they're all deficient for beta-carotene and vitamin E, right? So we wanted to know if we add, for example, beta-carotene to a cow's diet, can we increase beta-carotene concentrations in the follicular environment? That was basically a question, because beta-carotene can boost oocyte quality, can boost steroid production within a follicle. So that's what we investigated quite some years ago. So what did we do? And I will just show you the pictures. Don't look at the graph. I will just show you this. Look, if you give a dairy cow high beta concentrations in the feed bank, you see that follicular fluid concentrations goes up. And how can you see it? These two vials are follicular fluid taken from a dairy cow with ovum pickup. These two cows were supplemented with beta-carotene. And this is a control cow. So you can see that just by adding beta-carotene, the beta-carotene concentration in the follicular environment drastically increases. So yes, there is an opportunity through the feed bank to really change that microenvironment of an oocyte. Okay, last question. Can we implement preconception care? So you have an obese mother mouse or mother human, and future mother, and that person wants to become pregnant. Can we advise that person to, for example, lose weight for some weeks? And is that weight loss good enough to recover oocyte quality again? If we speak to gynecologists, they don't know. They all say, yes, weight loss is important. But if you then ask, is oocyte quality improving? We don't know. And then I asked, how much weight should a woman lose before she can think about conception? What's your preconception care advice? And their advice is really, it's not scientifically substantiated. So that's what we tried with mice. So what we did, we made mice obese and then submitted them to a preconception care intervention, as we call it. Or we put obese mice back on a controlled diet ad libitum, or we submitted them to a crash diet, right? And we wanted to study oocyte quality. So what did we do? We started the preconception care intervention with this experimental design, and every 2 weeks we sacrificed mice to study oocyte and embryo quality. Taken together, and just summarizing the results because everything has been published, applying preconception care through diet normalization is beneficial for oocyte quality. Lipid droplet goes down, metabolic health of the mother is improving, so that's really positive. However, we still see that mitochondrial features in an oocyte, they do not recover completely. They stay aberrant, they stay deviant. So mitochondrial aberrations, they stay even though the mother completely recovered metabolic health, has a normal weight again, etc. So that nicely links to what we see with the primordial follicle pool, that the primordial follicle pool is already affected by the diet. So to conclude, if you agree, to conclude, an unhealthy life hampers oocyte quality. I think that should be very clear. The status of obesity or metabolic stress, but also a direct impact of a deviating diet, are important. The impact can be very fast. The impact can be carried over to the next generation's oocytes. We know that the dormant follicle pool, which has never been focused on so far, is also clearly sensitive. We know that maternal obesity impacts offspring oocyte quality. There are strong opportunities to protect or even rescue oocyte quality, both in vitro and in vivo. We also know that preconception care strategies, at least in our animal models, they have the power to improve, but not completely recover oocyte quality. And yes, I'm convinced and hope you are all convinced right now as well that we should care about, and of course, definitely should care for the oocyte. Animal trials are confounded. So if you work with human or cow trials to focus on maternal diet and oocyte quality, it's very difficult to come up with clear-cut results because they are confounded, management conditions are there, they're underpowered. So it's absolutely not easy to come up with straightforward results. So with this, of course, I thank the whole team because they did the work. I'm just giving some orders. They do all the nice thinking and difficult experimental work. And of course, I thank you for your patience, your interest, for the invitation, and for your attention. Thank you very much.
Thank you very much, Joe, for the interesting talk. Now the floor is open for the questions. Yes,
Alireza,
please go
shoot.
ahead.
Suranga was first, but okay. Just thank you very much, Joe. It was very nice, and comprehensive, I have to say. And just thinking about Tom Fleming and the works that he has done, also Barker and all the, the hypotheses of the
Protein.
the developmental origin of the disease. So especially the fact that you're saying the mothers would also bring it to the children, and this is the reason also that we have the obesity that is ongoing. Also, evolutionary thinking, the idea is somehow the mother is giving cues and clues to the baby that, hello, prepare yourself for the world outside. And then the thrifty phenotype hypothesis is saying that the world outside is changing very fast. For example, in countries like China or South Persian Gulf, Middle Eastern countries, from a time that they have been involved with famine, now they have abundant amount of the material. Mothers prepare their babies to survive within very low nutritions, and now they have all this amazing food and so on. So hence, we end up with all these problems that they have with the diabetes and other things because being overweight and all. But in your case, you are someone saying that the mother is obese, and would that mitochondrial situation would also goes to the next one because that obesity has been there. So I am just wondering how, from an evolutionary biology point of view, we can explain this, and how can we say that the mother is giving the same phenomenon, the same disease, the same problem also to the next generation? Am I clear what I'm trying?
Yeah, you're absolutely clear. It's very clear. I will frame it a little bit differently. If the mother is metabolically challenged and unhealthy, which is now the new generation in the developing countries, as you said, they will jeopardize fertility of their offspring. Because what we did, we of course, mice are not human. What we did, we bought healthy young mice and we submitted them to an exploding diet, so to say. So we drastically, aggressively made them unhealthy and obese. If we have that condition, we see drastic impact on the next generation, oocyte and embryo quality. That's very clear. However, in the study of Ine, where we had maternal obesity and offspring diet, so we had maternal diet and offspring diet, and we did a crossover design. And it's absolutely very clear, Alireza, in our data, and I didn't expand on that, that if we have a mismatch, and there we go, maternal obesity and controlled diet in offspring, or maternal control conditions and offspring obesity, we have the biggest issues. So if there is a mismatch, we have the biggest negative impact. And that nicely fits in the thrifty phenotype hypothesis, where normally an offspring is prepared for the world he or she will live in later on. So we even see that a pup born from a control mother, from an obese mother that is then put on a controlled diet after weaning is not doing very well. They are not doing very well. We see that they are doing worse compared to the pups that we keep on the obese environment. right? So you're absolutely right. DOHAT is a very nice, interesting concept, but the drifted phenotype is probably more important. That's a first point I want to say. Second, please don't forget that we focus on oocytes in this perspective. And the oocytes of the F1 generation is already present in the uterus of the high-fat, high-sugar-fat mother. So apart from the genetic and epigenetic changes, etc., I'm sure that there is a direct exposure there of that primordial germ cell and then the primordial follicle within the fetus, within the uterus, within the obese mother. So all these cell systems are exposed to that toxic environment with downstream direct consequences. To really answer your question, we should look at F2 and F3 because we only focused on intergenerational effects. And to have the transgenerational effects in our model, we should go to F3 because F2 is still intergenerational. Because the oocytes is exposed within of F1 is exposed within the uterus of, so there is still a direct link. The reason why we stick to that model is that because we really want to mimic what happens in most societal contexts. If a young lady is born from an obese woman, there is a high chance she will be exposed to an obesogenic environment because she will be exposed to the same feeding habits. She will also be exposed to the same physical activity habits of the mother. That chance is very high. So that's basically where we focus on. But there is more going on. We, at the moment, are starting a project where we only focus on mitochondria. And we link mitochondrial DNA, we focus on mutations, etc. And then it becomes, of course, a completely different story. But that's too early to speak about that in detail. I don't know whether that's a little bit an answer to your question, Alidis.
Yeah. We can speak about it later also when maybe we meet each other. Thank you very much for the nice presentation. Thank you.
My pleasure.
Suranga, please go ahead.
Thank you very much, Joy. It was a very nice presentation. I really enjoyed. And I agree that we have to care for and care about. So that's no doubt. But I have a couple of questions probably.
Okay.
Would you think that, let's say in an ART setting, since we mostly go for ovum pickup, and can it be used as a biomarker like a NEFA or PUFA or MUFA, whatever the combination, in the follicular fluid as a readout or something to predict the oocyte quality? Or have you ever come across such anything?
You mean in a human, Suranga, or in an animal?
House, even in the commercial, IFPT setting or embryo production setting.
Yeah. It's a very good question, and there's a lot of literature about that.
I've seen, but nothing in practice. That's what I'm saying.
Is it
because that you cannot set a threshold for these MUFA, PUFA, like all the fatty acid, like?
You're absolutely right. So you may measure NEFAs, for example, but don't forget that they are very biolabile. So it means that if you stress your patients just during ovum pickup, you may have already a biased result. Also in the follicular fluid, once we did research on that, but it's a long time ago, where we punctured follicles to see how long it takes before the NEFAs from the blood can into the follicular fluid, and it goes really quick. So NEFAs are difficult from a biological lability point of view, but there are many other factors. So I know palmitic acid is known in human follicular fluid to be a marker for oocyte development capacity, but there are many others. The quickest these days that are used are quick genomic evaluations or gene transcription evaluations of cumulus cells, and there are some target genes you can use. Of course, it will take some time. And in commercial settings, as you know, every oocyte that you can reach will be submitted to maturation and fertilization. So if I speak to breeding companies, or if I speak to human assisted reproductive centers, and we start thinking about the marker, and I say, guys, do you want to design a project with me to design a marker? They say, and I will be a little bit blunt here, we don't give a shit. We take every oocyte we get and we fertilize everything. And if we are an embryo, we transfer. And then I say, yeah, but what about mitochondria and next generation? And they say it's too complex. Shut up, you're killing the business. So they don't want to pay me. That's the reason why I will have to come to Estonia to find money, I think, to do that research.
Okay, we would love to do something like that. And I have a second question about your oocyte pool thing. So it's known that, AMH is quite a good readout of, predicting or telling about the, primordial and primary pool of ovary. But what is the connection between this AMH plus, let's say, the fatty acid? Or what have you ever evaluated in your settings, like even in mice, even in cattle?
No,
Yes.
and that's a very interesting point. In mice, AMH is not so predictive for
Yeah,
the
but as
reserve.
cattle, yeah, but in sheep, of course, you can. In human, they
Yeah.
use it as a marker.
Yeah. It's a very good question. What we still have to do, but I still look for personnel to do it, to see whether our maternal obesogenic diet and offspring obesogenic diet, so the last model I showed you, has an impact on follicular depletion. That's basically your answer. Because it's extremely interesting to link it to human premature menopause or too early menopause. That's basically your question and the pathological impact. I still have thousands of ovarian slides available, but I still look for somebody who wants to start counting follicles, and that's not a very happy job. AMH and mouse is not so interesting. We do know, and we do hypothesize, and of course it's not a straightforward answer, but what we see in our primordial follicles linked to oocyte mitochondrial features, so these TFAM differences, PGC1 alpha and other mitochondrial markers, we think that this may have an impact on premature activation and premature depletion due to the maternal background. So that's our hypothesis. However, it's not so easy to design a nice experimental design to prove it. So your question is absolutely topical, super interesting. If you, I'm now digging in my head, in my memory, if you focus on AMH studies in bovine linked to diet, for example, or growth rates, puberty, these studies are not so straightforward. That's what I can recall.
Okay, thank you very much. I really
You're
enjoyed
more
it.
than welcome. It's my pleasure.
So yeah, I also have one question.
Mhm.
Now the trend is like females, they go for these keto diet plans. Keto means like you more fed on the fatty acids or fat food. So to me, I felt like after listening to your talk, like it's not a way to go.
No.
So do you have anything to tell about that?
Yes, I can be very straightforward, and Alirisa will agree, I hope. I think I'm a physiologist, and physiology dictates the rule of physiology is very simple. And my what I'm gonna say is not really scientific, but physiology, the central theme in physiology is do normal. That's it. If you go for the extremes in all kinds of physiological parameters, you may expect impact. Whether the impact is a big impact or whether it's dangerous, that's something else. But from a pure physiologically spoken, we know that whenever basic physiological processes are not going as it should be in a patient, in a human being, in a woman in this case, fertility will be the first factor that will stop functioning. I give you an example, women that are saying, I will stop sporting, I will start running marathons, I will do. That body is perfectly able to do that sport activity. But the first thing we will see is that ovarian cycles will stop. So fertility says, no problem, if you want to focus on sporting, go ahead, we will keep you healthy. But fertility will be paused for a while. Yeah, that's the link between the somatotropic axis and the fertility axis. They have at many levels crossovers, and that's how Mother Nature works. So if you suddenly change your diet from a well-balanced diet to a ketogenic diet, which is a diet where you stimulate the liver to produce more ketones, so you want to burn more fatty acids, basically. You want to focus not on gluconeogenesis, but on ketones. That's nice. There are many good things linked to that. Sport activity, lactate production by muscle, Brain function, by the way, is stimulated by ketones. But ovaries, based on the results I had during my PhD, if you add BHB, beta-hydroxybutyric acid, or acetoacetate or acetone to your oocyte maturation medium, you kill the oocyte. There I need to stop. So it's all about the balance. And some women will be very good because it reshuttles a little bit their metabolic footprint, which boosts fertility. But if you ask the question straightforward, is a ketogenic environment and a diet good for fertility? I would say no, because you push yourself in a certain direction, and an oocyte is not made to survive in that direction. But it can be very good for liver function, can be very good for sporting activity, can be great to lose weight, etc. By the way, losing weight will mobilize a lot of lipid-soluble toxicants as well, but that's another topic, of course.
Thank you very much. So is there any more questions to show? Oh, yeah, we have one question. So let me read out. Why is the implications of ER mitochondrial contacts in oocyte biology in the context of reproductive health? For example, how increased or decreased ER mitochondria contacts is associated with mitochondrial activation during folliculogenesis?
That's a very, very, very good question. We would like to focus on that as well, but I don't have the answer. I have to be very, very clear there. We know that endoplasmic stress, endoplasmatic reticulum stress markers are increased in oocytes from metabolically compromised donors. We didn't do any work in that direction. But in Australia, Rebecca Ropker did quite some work in human oocytes and mouse trials. But your question is really interesting because there you want to know the synergy between mitochondria and endoplasmatic reticulum. That's a really interesting question. I have no idea. We didn't focus on that. We may revisit our transmission electron microscopy features. So we have a lot of pictures of oocytes. So you're more than welcome to come and investigate all these transmission electron microscopy pictures to try to answer that question. I think I think the data that we have the material, but we need somebody who really wants to study study these pictures. Yeah, very good question.
Thank you.
Thank you very much. And if there's no more questions, I would like to say thank you very much for your time and interesting talk. And it's really amazing, quite a lot of knowledge to think forward.
That's true. It's challenging. Thank you very much.
Yeah, thank you. And just all participants, thank you very much for joining with us, and stay tuned with us for the future talks.